I'm Michael Betel, President and founder of the Fatty Liver Alliance, and with me today are three very special guests who will tell us about their exciting work in metabolic liver disease and how what they have demonstrated in the LiveWell study using hepatocyte can significantly reduce the time it takes to find patients who need follow-up and care. I'll start by introducing Dr. Tim Jobson, who's the medical director of Predictive Health Intelligence, Patrick Short, CEO of Sono Genetics, and Louise Campbell, founder and clinical director at Tawazun Health. Mass trial recruitment is one of the most challenging bottlenecks in clinical research, and early identification is one of the biggest gaps in liver disease. Your LiveWell study just out seems to address both. What do you think is the most important thing about the study design and the work that you're doing? And let's start with Tim.
So thank you very much, Mike, for having us on. So yeah, this is a really exciting time for us. We started the LiveWell study a while back, but have just been recruiting patients since March 2025. It's a diagnostic study looking at risk stratification using the hepatocyte tool at population level. So we're out there in the general population, looking at polygenic risk scores and looking at outcomes as defined through FibroScan, VCTE, and CAP scores. So that's what's been happening in the study. And what we did very differently in this study was use hepatocyte to risk stratify and then go out to recruitment. So we had special regulatory approval to approach the study in this way and just writing directly out to people saying, would you like to be part of a diagnostic study? And the response was incredible. So it was going to be multiple sites, but in the end, we just ran it in Somerset. The recruitment requirement, we were targeting 1,000 patients, and we've just closed recruitment. And yeah, we got there. So 996 patients recruited and consented to this study in just 10 months, which is a fantastic result and well exceeded our expectations when we started recruiting. So that's really exciting because it means we can reach out to the general population and bring them into a study, but also we've already defined some of their risk score. So that's great.
Yeah.
Um, the other thing, just to get this is much wider than just an investigator like myself recruiting people in a slow trickling clinic. So that's a very selected group of people. What we want to do is at population level and be much more inclusive. So people from deprived areas, people who have not had much contact with healthcare providers are coming into a study when they normally wouldn't be recruited.
And I know that we've been talking for years, about 4 years now, about hepatocyte and how things have progressed with you. And I love that you've partnered with Patrick's group. And I'd love to hear more too about the genetic aspect of it. I know almost everybody agreed to genetic testing, which is incredible. So maybe you can talk about that combination a little bit.
Yeah, thank you, Mike. That's right. I was pleasantly surprised with the high uptake of genetic testing. We run genetic studies across a range of therapy areas, but this is one of the first times that I think it's been done systematically at a population level in liver disease like this. So I was really interested to see the data and I was really surprised with the uptake. If I build on what Tim was saying, the strength of this program, I think, is that it uses existing health system data as the foundation. So rather than requiring a brand new test or some kind of esoteric new method, it starts with routine data collection that's happening in health systems. We did this in the national health system in the NHS in the UK, obviously, but I think this paradigm could be applied anywhere, starting with the routine health data and then layering on top this, novel biomarkers like the polygenic risk scores that you mentioned. We know that most patients with liver disease find out it way too late that they have it. And if we layer genetics on top of that, we know that most people don't have access to genetic testing today. So I felt like this was a really powerful, proof of concept that we could take this health system based approach to find patients early and also enable precision medicine trials in PNPLA3 in the future and other genetic subtypes of liver disease that we know patients progress more quickly as well. So it's even more important to find them earlier and get access to treatments or trials.
Yeah, at conferences around the world, people are talking more and more about genetic testing, and the comment does come up that it's not readily available in the general public. So it's an amazing, really a breakthrough that you're offering patients that they wouldn't normally have had. So that's a congratulations on that too. And Louise, your expertise in transient elastography, how does that play into all of this now?
I think for the study, this was the only interface that the patients had or the participants had with real-time information. Obviously, Predictive Health have gone through and read the results. I'm blinded as to why those patients were selected. And surprisingly enough, most people felt they were just the neutral patient and they were just there selected. So everybody assumes they don't have disease, which was interesting. But I get to tell them and talk to them about their liver health on the day of that scan. I can't change the pre-testing that's been done, neither can we change the genetics that have been done, but it's the one part of the study where they got real-time information and a discussion on liver health. And the engagement that we got, or I saw, because it's the only part of the study that I participate in, was amazing. They were very, very well engaged. There was a lot of high populations from around the area that had travelled and were prepared to travel. But some of these scans were arranged and booked within 2 or 3 days. So this ability for them to get real-time information when they wanted it in the location that they wanted was vital because their attendance was nearly 100% throughout, except during the winter when you get a bit cold. So for me, it ticks all on my boxes, as you know, Michael. It is about patient communication, offering solutions and the start of progress, but also not overly alerting them to something that's a panic moment. Because, of course, we were going to find high levels of disease in people who didn't actually expect to have it.
So thank you for that, Louise. So if the engagement rate is so high, I mean, I think that's pretty exceptional when you get anything over 90% on something like this, and what you've shown is scalable, then what's the future here? How can this be applied in a broader way to impact more patients?
So I think that one of the challenges in lots of liver studies is recruiting sufficient number of people for a prospective study. So there are large studies that are retrospective studies, but the number of high quality prospective studies that are recruiting enough patients who we know are out there, this is an extremely common condition. So our approach, I think, is applicable to lots of different studies. So whether it's an intervention study or a diagnostic study, risk stratification at population level leads us out there to the right patients. And what we demonstrated is we can get that engagement, we can get people in and get them enrolled in studies. So I think this is potentially a really powerful approach for. There's going to be more studies around specific genotypes. Again, we can do that, really easily now. So we've got 1,000 patients with genotype now, but we're moving on to we can do this again. We can replicate this in other sites very easily. So as we roll out hepatocyte, opens up more and more populations to these sorts of studies, and shows that large scale recruitment is feasible and effective. I think it's really exciting times ahead.
Yeah, it sounds like
It,
it. Yeah, Patrick, I want to hear from you too, and maybe if you can you share how many different genotype like tests you're doing?
yeah, so we do a genome-wide array, and that allows us to look at effectively the whole genome. We look at common as well as rare single nucleotide changes and small deletions across the genome. The only thing that we will not be able to get is ultra rare private variants, but this allows us to cover the common risk factors like the SERPINA1 gene, so the ZZMZ variants that are behind alpha-1 antitrypsin, also the missense variant I148M and PNPLA3, and also protective variants. So we'll share the full gene list, but it's very comprehensive, the approach. And Tim mentioned polygenic risk scores, so we're also able to look at combinations of genes and add to the growing literature of where when you see multiple risk factors or maybe a risk factor and a protective factor in combination, how does that change either risk of developing disease or progression or both? I also thought this study there is somewhat of a myth. I think that patients in this population aren't engaged in their health and that can make trials challenging. And I thought this is a really good myth-busting opportunity, both on the interest in genetics, but in the interest in taking part in something like this already. Louise mentioned spending 30 minutes with these patients to do a scan. I know that you would know that firsthand, that people are really engaged in this process. But I think this also shows that at scale, in a broad population, people are really interested in understanding more about their liver and metabolic health, and doing things that would set them on the right track. So for me, it made me optimistic about rolling these strategies out to clinical studies more broadly, because I think we know that patients will, if we lower the barrier of entry and make it easy to get a genetic test at home, which is how it worked in this case, to come and do a non-invasive FibroScan, and do so with the convenience of booking in a time and doing it when they can. If we lower that barrier to entry just a little bit, then people are really excited to participate in these kinds of programs.
Okay. That's great. And it's exciting, and it's really the future that we keep talking about. So it sounds like it's actually here already, which is really cool. A lot of people would want to know, what would the next step be after you've identified these patients? What, I don't know who wants to grab that, but
So I think there's two answers to that. So people who are identified with progressive liver disease, who are progressive fibrosis, who need intervention, clearly we need to bring those people into the appropriate clinical care settings. And one of the outcomes of any study like this is that you'll identify people who didn't know they had significant liver disease. I think where we need to go in terms of when we report this study, we need to be looking at how to help healthcare systems identify the missing patients. And we know the number of people out there with significant liver disease is high. And if we can use pre-existing data and then combine it with, as Patrick said, very easy to perform tests, at home or in the clinic, we're starting to see potentially a route to population level patient identification and risk stratification. So that's really exciting. That's kind of the clinical care aspect. In terms of trials, though, this opens up the number of potential participants hugely. Liver trials tend to take a long time. Well, lots of trials, all these trials tend to take a long time to report. So if you can recruit quickly, you can complete the study quickly, and you can get the data out there for the liver world to use. So I think that's super important. And using our methodology for intervention studies as well, identifying the right people, so that sponsors with new assets coming into the market can get those tested rapidly and get those trials completed. I think that's really exciting. So then we will have the therapeutics coming through. The therapeutic pipeline will be accelerated. And that's another exciting part of the future. So that's where we want to take this.
well, combining the opportunity to improve drug development and patient care is kind of a winning combo for everybody, right? So thank you very much, all three of you, for taking time out of your busy day to share this really exciting news. I know at the upcoming conferences we're going to learn more about the success, and we'll be following you in the next years to come to see where this plays out. So thank you very much again.
Thank you.
Thank you,
Michael.
Mike.